PhD candidate at Northern Arizona University, and I study riparian systems. What is a riparian system? Well, this.
See the stream down there? Even if you can't see the water, you know there is a stream there by the difference in the types of plants. We sometimes call these riparian systems ribbons of green because the plants that grow in the riverbanks are the only green things in a desert landscape like this.
This is a cottonwood tree. Cottonwood trees are one example of foundation species. Foundation species create locally stable habitat and provide ecosystem services for other organisms in the environment. Look, this cottonwood was felled by a beaver. Beavers use cottonwoods and willows to build their dams. Dams make ponds, and ponds create habitat for birds, insects, bacteria, fungi, other mammals, other plants, all sorts of things. Without cottonwoods and willows, the entire ecosystem would collapse. Riparian ecosystems take up only about 10% of the land area in the southwestern U.S., even though they support 80 to 90% of the biodiversity here. There's a lot of threats to riparian systems in the Southwest. We're only going to talk about a couple, but I bet you can think of more.
Such as dams and other ways that humans alter water flowing through streams, like diverting it to water crops.
The one that we're going to focus on today is this. This is a tamarisk plant. It was intentionally brought over from Asia in the mid-1800s. It was brought for erosion control, to provide shade for railroad workers, and hey, it does have pretty flowers. So why is tamarisk a threat? By the mid-1960s, most rivers and streams in the Southwest had dams built on them. People were also diverting water away from the rivers and streams to provide water for their crops. Now tamarisk, because of its extremely deep roots and greater drought tolerance, was able to do better under these hot, dry conditions than native trees. Tamarisk is now the third most abundant woody plant in riparian systems in the Southwest. Some land managers want to get rid of tamarisk. There's a lot of ways that we can do that. We'll talk about just a couple of them.
Managing tamarisk with chainsaw crews has its costs and its benefits. It can be really effective at a local scale, but it is very labor-intensive and expensive. Sometimes when tamarisk is removed with chainsaws, other non-native species move in.
This phenomenon is known as secondary invasions. We remove one invasive species, and a second invasive species moves in.
And then,
what do you do with all of this? The best way is to burn it. And that returns all the nutrients that are trapped in the wood back into the soil. But sometimes burning isn't possible. If there are archaeological artifacts at the site, then burning's not permitted. Sometimes these slash piles are left and become wildlife habitat. Just because you cut down the above ground portions of tamarisk doesn't mean that the roots are dead. Tamarisk is notorious for resprouting. So land managers will usually apply a herbicide to the stumps, and that's usually effective for preventing resprouting.
Researchers at NAU partnered with Babbitt Ranches to restore a stretch of the Little Colorado River. Our funding from the Pulliam Trust and the Wildlife Conservation Society helped pay for chainsaw crews from the American Conservation Experience to remove tamarisk in and around the restoration site. A crew of 8 people worked 8 10-hour days to clear 10 acres of tamarisk at this site. Between chainsaw removal and herbicide treatments, the ACE crews have controlled about 70 acres in and around this site. All that work has cost about $120,000.
Do you think that chainsaw crews are going to be an effective way to manage tamarisk?
Even though manual removal is effective at a local scale, like at the restoration site, tamarisk is found in every state west of the Mississippi River. So using a chainsaw isn't going to get rid of all the tamarisk anytime soon. And lots of places tamarisk exist are not accessible by chainsaw crews and heavy machinery.
We need a special force of biological control agents.
This is a tamarisk leaf beetle, and it is a biological control agent of tamarisk. This little beetle's ancestors were brought to the U.S. from Asia. For decades, research was done to make sure that they would be safe to be released into the environment. In 2001, the tamarisk leaf beetles were released in 6 states in the western U.S. by the USDA's Animal and Plant Health Inspection Service. We call it APHIS for short.
Those little beetles can defoliate a big tamarisk stand like this one in as little as 2 weeks. This is great! What could possibly go wrong?
See this cute little bird? This is the Southwestern willow flycatcher, and it is an endangered species. This bird species needs to have standing water near its nesting sites. In many parts of the bird's range, tamarisk is the only tree available to nest in.
When APHIS released the tamarisk leaf beetle, they knew that the Southwestern willow flycatcher were nesting in the tamarisk. But don't worry, they said. It'll be fine, they said. The beetles can't complete their life cycle that far south, they said. Well,
if tamarisk leaf beetles eat all the leaves on a tamarisk that a Southwestern willow flycatcher is nesting in, the nest is likely to fail. Without leaves, the nest gets really hot, often so hot that the eggs or baby birds die. Also, a nest in a defoliated tamarisk is more visible to predators.
APHIS, I hereby order that you have to restore native plants after beetle defoliation in the Southwestern willow flycatcher range. In 2013, a lawsuit was filed against APHIS by the Center of Biological Diversity and the Maricopa Audubon Society for APHIS's failure to protect the Southwestern willow flycatcher from the effects of beetle defoliation. In 2017, a judge ordered that APHIS restore native cottonwoods and willows within the Southwestern willow flycatcher to provide habitat for the endangered bird. Now it's really important that we figure out how to turn this into this, but that's easier said than done. Restoring tamarisk invaded lands is difficult for many reasons. One of the things that makes tamarisk so successful is that it can tolerate salty soils. In fact, it increases the levels of salt in the soil by drawing up the salts that are way down deep, using its deep roots, and then it brings that salt up into its leaves, packs it into the leaves, and then when the leaves fall, all that salt is now deposited on the surface of the soil. Another way that tamarisk changes the soil is not a chemical property like the salt, but a biological property. Something that's in here, or should I say, is not in here. Tamarisk don't need mycorrhizal fungi, but cottonwoods and willows do. Mycorrhiza? Myco means fungus. Rhiza means root. So mycorrhiza literally means fungus root. Mycorrhizal fungi form typically beneficial relationships with most, but not all, plants. Here are some roots of a pine tree. They don't look like much, but when we look closer, I have the roots set up underneath that microscope over there, but to make it easier to see, we'll look here at this computer screen. Here's a root tip that doesn't have any fungus associated with it. So it's only plant root. Over here, though, we see a root tip that does have fungus associated with it. See how this one is swollen and is a little bit different color than the root itself? This is a mycorrhiza. This is the joint organ made of both plant and fungus. See over here? Here's another species that's associated with the same plant. See how it looks different? Different species of fungi provide different benefits to plant. This kind of mycorrhiza is called an ectomycorrhiza, and it's just one of the ways that plants and fungi make these types of partnerships.
Plants make sugar through photosynthesis. They provide some of that sugar to their mycorrhizal fungal partners. The fungi provide the plants water and nutrients from the soil that the plant otherwise wouldn't be able to access on its own. Both the plant and the fungus benefit from this mutualistic partnership.
Dry and salty conditions aren't good for native plants or their mycorrhizal fungi. Even though we don't know how, we do know that tamarisk reduces the number of species of mycorrhizal fungi present in the soil. The chemical and biological changes that tamarisk makes to the soil can last for years after it is killed by the tamarisk leaf beetle or removed. And that makes it really hard for cottonwoods and willows to grow, whether they're growing from seeds naturally or they're planted.
Not only do cottonwoods have to deal with tamarisk, they also have to deal with drought. Just like in 2019 and 2020, there was a long and severe drought in 2002. Some researchers took that opportunity to see how the amount of tamarisk relates to the number of cottonwoods that survive. Here's what they found. Along the x-axis is the percentage of land covered by tamarisk. On the y-axis is the percentage of cottonwood in that same area that died. As you can see, as more tamarisk was present, the higher the percentage of cottonwoods that died. But look at this, of all these populations represented by one of these dots, only one population had 100% mortality. That means that even though there is a bunch of tamarisk around, there are some individuals that can survive tamarisk and drought.
I'm going to call these experienced cottonwoods because they've either adapted or acclimated to living with tamarisk. I'm interested in learning why these trees survive and even thrive when others, like these naive cottonwoods, would likely suffer if not die if tamarisk invaded this area. Remember, there's both an ecological and a legal need to restore tamarisk invaded lands. I did a greenhouse study here at Northern Arizona University to find out a little bit more. I wanted to answer the question, how can we improve restoration outcomes in tamarisk legacy soil? So I collected cuttings from experienced cottonwoods, cuttings from naive cottonwoods, and grew them in the greenhouse with two different types of soil. I used tamarisk legacy soil and agricultural legacy soil. This is my control treatment. I also collected healthy soils like this that's full of those beneficial mycorrhizal fungi from sites where there's no tamarisk. Half of the trees received live soil inoculum and the other half received sterile inoculum. By microwaving the soil, it essentially kills all of the beneficial fungi and all the bacteria and anything else that's living in that soil. That's my control treatment. So what did we learn?
Here are the results of the inoculated plants versus the sterile inoculated plants. The live inoculated plants are represented by the black bar. The sterile inoculated plants are represented by the gray bar. You can think of the zero line as the soil surface. Anything above the zero line is the shoot mass. Anything below the zero line is the root mass. So when we compare both the roots and the shoots, we see that giving the cottonwoods live sterile inoculum increased their growth by a considerable amount.
Does adding mycorrhizal fungi and other soil microbes increase the growth of cottonwoods in tamarisk legacy soil? It sure does. The second part of my greenhouse study asked the question if experienced cottonwoods grow better in tamarisk legacy soil than naive cottonwood trees growing in tamarisk legacy soil. Well, let's see.
Let's look at the experienced versus naive cottonwoods. When we look at this graph, what we're looking at is experienced and shown in yellow, the naive shown in purple. The zero line you can think of as the soil surface. Anything below the zero line will be root biomass. Anything above the zero line will be shoot biomass. Let's look at the shoots for a minute. The experienced cottonwoods and the naive cottonwoods had the exact same amount of shoot biomass. But when we look at the root biomass, we see that experienced cottonwoods grew more roots than naive cottonwoods.
Do experienced cottonwoods grow better than naive cottonwoods when grown in Tamara's Legacy soil? They sure do. But what happens when we do both things together? We use experienced cottonwoods and we add mycorrhizal fungi. What will happen then? Will experienced cottonwoods respond differently to the addition of mycorrhizal fungi than naive cottonwoods? Take a minute or two to think about what you expect to happen. Your teacher might decide to make this a homework assignment. So write down your answer with your justification. Pause the video. I'll be here when you get back. I predicted that experienced cottonwoods wouldn't respond very much to the inoculation because they're already used to growing without mycorrhizal fungi. We can calculate the boost that the plants get by adding the mycorrhizal fungi by comparing the inoculated plants with sterile inoculated plants. And that gives us a number called the mycorrhizal growth response. So when we look at our mycorrhizal growth response graph, what we're going to see is a y-axis and an x-axis. Along our y-axis, we always put the thing that we measured. So in this case, it's going to be our mycorrhizal growth response, our MGR. Along our x-axis, we have our different treatments. So for this example, I'm just going to put T1 for treatment 1, T2 for treatment 2, T3, and T4. So far, this looks pretty normal, right? But I'm going to add something now that we might not always see. Sometimes we see it, and it's a zero line. Okay? Anything then above the zero line is going to be a positive value, of course, and anything below is going to be a negative value. But what the zero line represents is the average of all the plants that were given the sterile inoculum in these different treatments. That's their average. So we can think about this as the sterile control. Okay? So this allows us to see how beneficial those mycorrhizal fungi actually are. And so if our data crosses the zero line, then those plants that were growing in treatment 1 didn't get any bigger or smaller compared to our sterile control. There was no effect of adding those mycorrhizal fungi at all. So we're just going to put meh face. They don't care. Okay? So, but if we see a negative value, then by adding those mycorrhizal fungi, we're actually seeing those plants grow smaller than the sterile control. So instead of being beneficial, the mycorrhizal fungi actually were harmful. So let's put a little sad face here. But then if we see a positive value, then that means that the plants in this treatment grew bigger than the sterile control. That's what our zero line is, remember? And so we can say that yes, those mycorrhizal fungi benefited the plants. And that bigger the positive value, the more beneficial those mycorrhizal fungi are. So what do you predict? Do you think the experienced cottonwoods are going to get a bigger boost? Or maybe the naive cottonwoods are going to get a bigger boost? Let's see if you're right.
What we see is that both experienced and naive cottonwoods benefit from being inoculated with mycorrhizal fungi, but experienced cottonwoods benefit more than naive cottonwoods. This result was not what I predicted. What about you?
Experienced cottonwoods respond differently to the addition of mycorrhizal fungi than naive cottonwoods? Yes, they do. So what did we learn from this experiment? if we get cuttings from experienced trees like this and soil inoculum from sites like this, we'll be more successful when we try to turn this into this. But do the results of this greenhouse study actually translate to real-world situations? That's what you are going to help us find out. Several years ago, some researchers came and collected some cuttings from this cottonwood tree. And we know because they left a name tag on this tree. I know because I was one of those researchers.
NAU researchers have partnered with high schools to restore a stretch of the Little Colorado River near Cameron, Arizona. With the help of high school students, we have planted about 8,000 cottonwoods and willows at the restoration site.
High school students like you planted these cottonwood trees. Some of these trees are from experienced sites, some are from naive sites, some were given live inoculum, some were given sterile inoculum.
Now you get the chance to come here and continue the work that other students like you started. With your help, we will collect data on how well these trees are growing. The results of this experiment that you have the opportunity to help with will go much farther than an individual tree or even this restoration site. Our results have the potential to improve restoration outcomes across the western United States.
Remember, cottonwood trees are foundation species. The work that we're doing at this site isn't just about this tree, but it's about restoring the entire ecosystem. Inoculated plants with the not live inoculated plants. I'm going to cut that part so it doesn't matter. I forgot my line. I'm going to turn this into this. I think I was looking at the butterfly too. At this site, crews of 8 people. Okay.
8 people were 8. Oh boy.
Action. With cap. Try that again. Some of these cottonwoods are from experienced sites, some are from naive sites. I want to take my sunglasses off. Okay, last time. Ready? Here we go. 4. See this stump? This was chewed by cottonwoods. Nope. 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 59. The second part of my greenhouse study asked the question if experienced cottonwoods grow fat. To load all this up into a truck, take it somewhere else, burn it there, and then, I don't know. Find out, and I'm still holding my remote.